We all know that machines are powered by electricity, but you might not be aware that you are watching this video thanks to the electrical events taking place in the nerve cells of your brain. This electrical activity is initiated and regulated by your ion channels. Ion channels are a type of membrane proteins that allow the flow of ions and are found in every cell of our body. They govern every aspect of our behavior, but sometimes do not function properly, giving rise to the so-called channelopathies.
Many cardiac diseases are caused by electrical disturbances of the cardiac functions. We call that arrhythmias. And very often, ion channels have found to be causing these cardiac arrhythmias. However, we're still lacking a detailed understanding of these ion channels in normal physiology and in diseases. And we're currently collaborating with Dr. Medeiros Domingo from the Inselspital, who is seeing such cardiac arrhythmia patients with similar channelopathies.
Our research here in Inselspital focuses on the molecular basis of inherited arrhythmia syndromes. That means that we look for mutations or for novel genes that are associated with cardiac channelopathies or cardiac myopathies that sometimes can give sudden death or sometimes can give just long-term heart failure.
So the specific goal of this TransCure project is to understand the role of this TRPM4 channel in cardiac diseases, in multiple sclerosis, and as well as in prostate cancer. And as a team with chemists, biochemists, and physiologists, we are trying to develop small molecular chemical compounds that using as tool compounds to understand these diseases.
My work consists of characterizing mutations found in TRPM4 gene in patients with cardiac conduction disorders and to understand which is the mechanism leading to a misfolded protein in this kind of patients. Moreover, I'm also involved in a project consisting in characterizing TRPM4 in neurological disorders such as multiple sclerosis.
Here in our lab, we are interested in studying the pathophysiology of TRPM4 in cardiac conduction disorder. And I'm basically focused on studying the physiological role of TRPM4 using patch clamp techniques and also some of the drug screening facility in identifying the role of TRPM4 in this cardiac conduction system using cardiac myocytes and also even some of the hex cell lines which we have in our lab.
The research of this NCCR project helps and facilitates the work of Dr. Medeiros Domingo by giving her tools to better diagnose these patients. And on the top of that, we're developing small molecular chemical compounds that may be drugs in the future to treat these patients.